Now, on the first 2 pages of notes, we really were just talking about terms related to DNA, because one of the most important things that we have to divide in a cell is the DNA. So we were talking about what we call DNA when it's long and stringy. What does it mean if a chromosome looks like a single line versus an X? So now whenever I reference these molecules, you have an idea, a better understanding of what's going to happen if a cell gets a signal to divide. Now, most of your cells should not be in any stage of division, nor should they be attempting to divide. And whenever cells, because remember, cells are only supposed to divide when they get a signal. Whenever a cell dies, or you need to make new body cells so that you can grow and repair tissue, that's when cells need to divide. But cells do not need to divide if you do not need to grow, or you do not need to replace dying cells. So most of our cells in our body, like these upper parts of our skin cells, we're never going to see these cells divide unless you scrape your knee or you get a cut. Now, cells that are not actively dividing or preparing to divide, that are just doing a job for you, we say that those cells are in a phase which is called GO phase. Okay? Now, there's some cells that can never divide, even if the cell next to it dies. So, for example, let's see if I can pull this up real quick. Actually, I don't have it open yet, so I'm going to forego that. So, a muscle cell is an example. Let's write muscle cell. Let's write muscle and nerve cells. If you damage and kill muscle cells and nerve cells, the muscle cells that are in that area, or that nerve cells that maybe are next to that no longer functioning cell, they can't divide and give you new ones. So if there's nerve damage and someone is paralyzed, then the nerve cells in that area of the body cannot divide and give you new ones. And so paralysis is permanent. Now, we are trying to do things like use stem cells. Remember, stem cells can turn into any type of cell. So we're working on using stem cells to put in different areas of the body so that they can regenerate into cells and help heal you. Now, whenever a cell in GO phase gets a signal to divide, then we say that the cell enters the cell cycle. So here's a cell, and this cell is in GO phase. And let's indicate there's a receptor protein on its membrane, because remember, cells have receptor proteins. They can receive signals. They can communicate using their cell membranes. So let's say a cell next to this cell dies, and some of those chemicals from that cell are released. They can bind onto the cell, and now it's time for this cell to divide. So we say that the cell that was in GO phase is going to enter the cell cycle. Now, what I mean by that is this cell is now going to go through some different stages that we have named, and eventually it's going to split into 2. Now, I kind of think of these stages as being like stages in your life. So the first stage in your life after you're born is you're an infant, and then you're a toddler, and then you go through adolescence. And so all those stages are part of a bigger stage, which is your childhood, right? And then after you're a child, you become an adult. And so you just go through different stages of your life. So when the cell gets a signal to divide, we say that the cell is going to enter the cell cycle. Now, you can see there's different phases that are going to happen throughout the cell cycle. And there's 3 main phases in the cell cycle. So the first one is listed for you right here, which is interphase. So interphase, and I know this isn't color-coded, but interphase is all of G1, S, and what's called G2 phase. All right, so there's 3 main stages. We have interphase, then we end up with a cell going through mitosis, and then the last stage that the cell's going to go through is called cytokinesis. So let's indicate we have interphase, and then a second main stage is mitosis, and then we have cytokinesis. Now, how I like to remember kind of what's going on in these stages, well, first, instead of writing my I like that, if I just write it like that, that could be mistaken as a 1, right? So interphase, that I looks like the number 1. That's to help you remember that's the first main phase of the cell cycle. And really, this one is just, interphase is just preparing, getting ready to divide. So if you've ever moved, the majority of your time isn't actually moving your stuff. That happens over like 1 or 2 days. The majority of the time is just preparing, packing everything up, making sure you have enough boxes and people to help when it's time to move. So this cell is going to prepare to divide, and it's going to be doing that all throughout interphase. If everything is good to go, then the cell's actually going to divide. So really, these 2 stages down here have to do with the division part. Mitosis is the events that are going to divide the nucleus and the DNA, and then cytokinesis is going to divide up the cytoplasm and all the contents in there. So hence the name cytokinesis. Kinesis means movement. Cyto, cell, we're dividing up, we're moving around the cytoplasm and the organelles. As you can see from the diagram, interphase is a long stage, and it itself has 3 sub-stages, G1, S, and G2. We can call them G1 phase, S phase, G2 phase is fine, or G1, S, G2. And again, the cell is going to prepare. We're going to get into more details of what happens, but we're just going through a general idea of what's going to happen when a cell goes into and completes a cell cycle. Whenever the cell completes those 3 phases of interphase, then it's time to divide. And so to divide the nucleus, the cell's going to go through 4 stages. I'm going to write P, M, A, T. So the 4 substages of mitosis are prophase. So P, M is metaphase. A is anaphase. And then the last one is telophase. Prophase, metaphase, anaphase, telophase. So we usually abbreviate it. If you think of PMAT, or like a saying like purple monkeys at Taco Bell, that might help you remember the order of the stages of mitosis. Now, as I mentioned, that during mitosis, we're all about dividing the DNA. We're about dividing the nucleus. So that's why sometimes mitosis is referred to as nuclear division. Okay, remember that mitosis has TOS in it. So if this cell gets a signal and it divides by mitosis, we're going to end up with 2 of the same, 2 daughter cells that have the same number of chromosomes as the cell that splits, 2 of the same. By the way, before I move on, I want to make sure you know that this cell that is going to divide is called the parent cell. And whenever it's done, it's gone. Half of it now exists in this cell and half of it exists in that cell. And those 2 are called the daughter cells. Now, it can get confusing because if you have a parent and you have 2 daughters, you have 3 people. Well, that's not the case. A parent cell is going to end up becoming the daughter cells. So when this splits in half, we don't say it's a parent cell anymore. We say we have 2 daughter cells. Okay, so as we mentioned, 2 of the same. So be ready for scenarios that if I tell you this is a diploid cell that has, let's say, it's a species that's diploid and they have 40 chromosomes. If I say this cell divides by mitosis, how many cells do we get at the end? 2. If I ask you how many chromosomes will be in each, it's going to be the same as what it starts. So these daughter cells will end up with 40 chromosomes. They will be diploid just like the original cell that divided, which that's what we need. If this was a skin cell and the skin cell next to it died, we need this one to split and replenish back this and replace that cell that died. And we need it to be or have the right number of chromosomes in it. All right, so as we mentioned, 3rd phase, cytokinesis, is really just dividing up the cytoplasm. And the cytoplasm, remember, it holds proteins and all of the organelles. So cytokinesis is the division of the cytoplasm and the organelles into 2 separate cells. Now we're going to take a look at what this looks like, and we're going to be using different colors. So if you are watching this video, you're going to definitely want to go and get like 4 different colors of pens or pencils. I'm going to try to use blue, my blue's going to work. So I'm going to probably use blue and purple and red and just use like my pencil for black. So we need 4 different colors. So we're going to draw what it looks like if there is a cell and it gets a signal to divide. So let's just start off with a cell that would be in GO phase. So this cell is just doing its job. It gets a signal to divide. All right, so then the cell is going to move into the cell cycle. Now we're going to pretend this cell is supposed to have 4 chromosomes in it. So it's a diploid cell. It's supposed to have 4 chromosomes. When we're done, we should end up with 2 cells that are just like this, that are going to be diploid with 4 chromosomes. So once it gets a signal to divide, we say it enters the cell cycle. So now the cell is in G1. I want you to underline G, and we're going to write grow, because that's one thing that's going to happen a lot. The cell is going to start to grow. We're going to end up splitting it into 2 cells. So this cell is going to grow. It's going to make additional organelles. So why don't we put, a few like smoothie R and Ref R in there. Let's make some more mitochondria. Remember, mitochondria look like they have little squiggles in them. And the cell's also going to be manufacturing a lot of proteins that are going to assist in the next stages. So the cell's growing and making additional organelles and proteins. Now, something that is really important is the cell is also checking. It's the first time. So see that one? It's the first time the cell is going to check the DNA for mutations and repair it if needed. Okay. Now, what does the DNA look like as it's being checked? DNA is in chromatin form, and it only coils up into chromosomes during mitosis. So if this cell has 4 chromosomes, the DNA is just long and stringy and it hasn't coiled up. And that's what I like to diagram here. DNA is just in long molecule form, in chromatin form. And if this cell has 4 chromosomes worth of DNA, that's why I'm drawing it just with 4 different colors. So the cell has 4 chromosomes. When the cell is whenever it's copying its organelles, making proteins, the DNA is being checked. Nothing has really happened to the DNA. It's still there. It still looks like what it did a few minutes ago. And the DNA is in chromatin form while it's getting checked. Okay. Now, if the DNA is good, then the cell is going to move into the next phase of the cell cycle. And so next to S phase, I want to write synthesis. That means something is being made. So synthesis means to make. What the cell is going to do is it's going to make a copy of every chromosome. So I like to reference that, or I like to model that as, see my DNA? Just make this pile of string twice as big. There's twice as much. So there's twice as much of my purple and my blue DNA, my red, and now we have twice as much DNA. Okay? All right. Now, the cell, so S phase is all about copying DNA, making an extra set. And that's called replication, DNA replication. We'll study that in January. If you replicate a photo, that means you make a copy of it. So now that the cell has grown, there's extra organelles now. I'm just not going to draw them. The cell moves into G2 phase, and one of the most important things that is being checked, because it is a lofty task trying to build and copy every chromosome, is we're going to see if the DNA was replicated correctly. So again, the DNA, there's still twice as much of it. It's just being checked. So let's draw our DNA sitting here. There's proteins that are going to be checking and trying to find and fix mutations. The DNA is going to be checked after it's been copied. The cell also, again, is going to keep making additional organelles. So let's add, again, this cell's going to be got plenty of organelles. It's going to have lots of proteins to assist in division. And just putting some organelles in here. We need lots of organelles because we're going to have to divide organelles into 2 daughter cells. We also can underline this G over here to help you remember that also. You're going to see even from here to here, the cell grew some more. So more growth, the cell is going to grow as well. And collectively, these 3 stages, so that shows us what a cell looks like as it's moving through the 3 phases of interphase. So technically, these 3 stages are interphase. Now what can be confusing is sometimes we're going to have to draw pictures so you can see a cell and how many chromosomes are in it. And the thing is, this cell doesn't ever exist. You can't find a picture of a cell that just has DNA in chromosome form and it just looks like single lines here, because you can't ever see chromosomes until after the cell copies those chromosomes, which we're going to draw that cell here in just a second. But if you see a picture like this, that just means that we have a cell. It's a diploid cell. It has 4 chromosomes. They're just trying to show you that by drawing them like this, even though this is not really what the DNA is going to look like. It's just going to look like this chromosome is going to be uncoiled as chromatin. Same with that one and same with that one, okay? Now, if you were looking at a cell in a microscope that's in G1 phase or in GO phase, it won't look like that. It actually won't look like this in the microscope, because in the microscope you can't see DNA. You can't see DNA when it's in chromatin form. And so cells that are in G1, S, and G2, all 3 of them, they just look like this. The DNA, it's in chromatin form. So let's just show that there is DNA in here, just lots of stringy DNA, but you won't ever see it. You're just going to see a cell. You're going to see a dark circle in the middle with that's the nucleus. And so you can't actually see the DNA using a microscope looking at cells, because the DNA is in chromatin form. So sometimes I am going to show you some real cells and what they really look like. And so when you just see a cell and it looks like this, we're going to say, oh, this cell is either in GO phase or it's in one of the interphases stages. It's maybe in G1 or maybe it's in S or in G2. You can't tell just by looking at it, because it just looks like a cell with no DNA that we can actually see in the nucleus. All right, G2's done. So now it's divide time. So it's time to divide. So now we say the cell moves into mitosis. Remember, there's 4 stages of mitosis. PMAT. Now, you will need to be able to tell me what is going to happen in the different stages. And there's a lot that goes down in prophase. Now, every single, the set of copied chromosomes are going to coil up into chromosome form. So this is the first time you can see DNA, and we can even see this DNA now if we're looking at the cell underneath the microscope. So that pile of red chromatin is going to condense, and now it looks like a red X. And the pile of blue is going to condense into a blue X, and so on. So our DNA is going to coil up into chromosome form, and we can see it for the first time because it's much thicker now when it's in chromosome form. Now, we also are going to have to move our DNA molecules around, so you can see that the DNA is going to get moved to the middle. So we need to get rid of that nucleus because it's a barrier. So one thing that happens in prophase is the nuclear membrane disappears, and that's what we're showing you here by this dashed line, because this is the nucleus. That nuclear membrane is going to start to break down and disappear. Now there's some structures that are going to help push and pull these chromosomes around in the cell. So let's label these 2 little cylinders here and here in the middle. Those are called the centrioles. I used to teach them when we talked about cell parts, but you already needed to know many cell parts, and so I've kind of gotten away from that. I'm just teaching about the cell part here in cell division, because that's when the centrioles are going to be used. So the centrioles, you can see that from the centrioles, we have these little fibers that are starting to form and grow. And eventually, they're going to reach out and they're going to hook on to our chromosomes. Remember these chromosomes? They look like X's because they were copied. And these fibers that are forming here, that you see on both sides, we can call them the spindles, or lots of times they're called spindle fibers. So we say in prophase, the spindle fibers will start to radiate out from the centrioles. You can say the spindle fibers will start to grow out from the centrioles. All right, so let me recap. Prophase. DNA goes from chromatin into chromosin form. The nuclear membrane is going to disappear. Centrioles will start to move to opposite sides of the cell, and the spindle fibers are going to start to grow. And eventually they're going to hook on to these copied chromosomes, or you can say the sister chromatids. Now the next part's going to get real easy. Whenever these chromosomes get pushed and pulled and lined up in the middle, then we say that the cell is in metaphase. If you remember, M for metaphase, M for middle. When the chromosomes are aligned in the middle, we say the cell has reached anaphase. Metaphase. Getting ahead of myself. The next phase, you can see our chromosomes are no longer as X's. They're getting pulled apart. So one side of this chromosome, so one sister chromatid is going to be pulled this way, and the other sister chromatid is going to be pulled the other way, and we can see that happening there. Now, let's color code this so we make sure we understand what's going on. So these chromosomes are just now going to be lined up in the middle. So let's make sure we have each chromosome lined up in the middle. And then in anaphase, those chromosomes are getting pulled apart. So this black chromosome is no longer in X form. It got pulled apart. And then my purple chromosome, now half of it's going one way and half of it's going the other way. The blue chromosome gets pulled apart. The red chromosome gets pulled apart. And now the two copies are no longer attached together. The sister chromatids have been separated. So we're going to remember A for anaphase, A for apart. When the sister chromatids get pulled apart, we say the cell is in anaphase. All right, last phase, telophase. So we went through P, M, A, T. If you remember what happens in prophase, the opposite is going to happen in telophase. So telophase, we're going to remember, is the opposite of prophase. So remember in prophase, our DNA was in chromatin form and then coils up into chromosomes. Well, now our chromosomes are going to start to unwind back into chromatin form. So that's what we're trying to show you by adding that these chromosomes, if there's like a little thread, you can pull them. They would just kind of like, they would just unspool, or they would just go back into string form. Our nucleuses are starting to come back. So let's just underline the nuclear membrane. So it looks like we're getting a nuclear membrane around this set of DNA and around that set of DNA. So the nucleuses are coming back. The spindle fibers are starting to disappear. So we don't need them anymore. And we now basically have TOS. So remember, our original cell had 4 chromosomes in it. So now this daughter cell over here is going to have a red chromosome. So will this one. It'll have one blue one. It'll have a purple one in each. And we've got then the black one. All right, let's go ahead and talk about the third phase of the cell cycle. Now, really, telophase is just what's happening to the DNA in the nucleuses. But during telophase, the third stage of the cell cycle is already starting, cytokinesis. The cell's going to have to split the cytoplasm into 2 daughter cells. And you can tell the cytokinesis is already also starting to happen in this cell because of these indents that we see. That's part of cytokinesis. So in animal cells, the cell membranes are just going to be pinched and pulled in until they meet, and eventually you're going to have 2 cells. Now, this indent is called the cleavage, a cleavage furrow. And we see it here, and we see it here. Actually, I don't want to draw that there. So we can put cleavage furrow here and here. I'll show you another picture of it on the next page. All right, and so the cell is going to split the cytoplasm kind of evenly here and the organelles between the two daughter cells. Now, let's go ahead and draw our two daughter cells. So now that they have been split apart, let's draw two daughter cells right here. Each one has a nucleus, because the nuclear membranes come back, and this one is going to have a red chromosome, this one will have a blue, this one will have the blue, this one will have the DNA from the purple chromosome, and then from the black one. And our daughter cells, two of the same. They look exactly the same as the original cell that got a signal to divide. So TOS, two of the same. All right. One little part of the next page, and then we'll pause for this lecture. As I mentioned, animal cells, the plasma membrane is going to be pulled in together, and then it'll meet, and eventually our two cells will be completely divided from each other. And that indent is called a cleavage furrow. So cleavage means to cut in half, and if you look at a real cell that is dividing, this is what a cleavage furrow looks like. So it's also happening on the other side. So the cell membrane, the plasma membrane, is being pulled towards into that cytoplasm. So the cell can cleave, which means split in half, right? Plant cells, though, they have an issue because they have a cell wall. So they can't just pull in this wall because it's rigid. It's not going to go anywhere. It's a strong structure. So the plants are going to build a cell wall and cell membranes in the middle, and they're going to use the post office of the cells. So if you remember, the Golgi are the post office of the cells. They're sending vesicles, so these little circles are the vesicles being walked along cytoskeleton. These vesicles are being sent to the middle where they will meet, they will fuse together, and if you remember, they're made of phospholipids. So the phospholipids come together to form a bilayer here, and between these two phospholipids bilayers, we will get the contents of the cell wall dumped, because that's what's in the vesicles, is the cellulose and enzymes that are going to be used to build the cell wall.